you guys, 1 second. Okay. Yes. Good afternoon, everyone. Yeah, I think everyone completed the B4 chapter test, and as I know, like some of you already told, right? It's good. Some of you attempted very well. We will see the results. Okay. And this week you don't have any chapter test because for third and fourth chapter, sorry, third and fourth batch, the that particular chapter is not yet over. Okay. So probably the cell chapter test will be in the next week. Okay, so we'll see that. And another one thing, guys. What I want to mention. So complete the modules. Okay, I don't know how many of you completed the cell modules. So complete it. And if you have any doubt, just put the message in the this one, group, so I can also answer, or somebody else also can answer. And remember another one thing, in the module, sometimes what happens, the answer may be wrong, key answer may be wrong. Okay, why that happens? Because they are in the series, they are giving A, B, C, like that, right? Sometimes misplacement will happen. That's why. Okay, so if you are damn sure about the correct answer, and if you still have that, is it right or wrong, then you can ask me, or you can discuss with your friends also. Okay. Good afternoon, Anu. So we can start, guys. So new chapter, biomolecules. We are lagging in 2 classes because of some issues. My electricity issue one day, so little lagging, and another one is like my health issue. So I think everything is sorted now. Good afternoon, Naina. Okay, so after long gap, we are back to the classes, right? So first, what we have to do. Good afternoon, Amrita, Raksha. So first thing we have to do is like start solving one or two questions, right? Yeah. Good afternoon, Vibha. So first question came. Nitrogenous waste is excreted mainly as. The question is simple. Nitrogenous waste is excreted mainly as urea in both frog and tadpole, urea in frog and ammonia in tadpole, uric acid in frog and urea in tadpole, urea in tadpole and ammonia in frog. Okay, just think about it 1 second. Sorry, take 10 or 13 seconds, then try to answer it. Okay. Good afternoon, Gopika Ashwin. Yep. I'm just launching the poll, guys. I think you guys already read the question. Question is easy because it's mostly based on what we studied in the B4, and you already read B4 for the chapter test. So I don't think it's tough for you guys, right? And Ashwin, Gopika, and Anu, how is the chapter test? I just want to ask you guys also, like, did you guys attended, or if you attended, how is the test? Okay, I'm getting answers, but like, okay, it's a mixed response, no issue. Okay, you guys, good. So last 20 seconds, we'll see. Anand, check one more time. What? Result will come, no issue. Result will be given in like 2 to 3 days. Yeah. So I'm ending the poll, guys. So I think why there's like so many less people attended the poll. It's an easy one, right? I don't think it's a hard one, right? I'm ending the poll. Good afternoon, Srini. Yep, I'm ending the poll and I'm sharing the results. 4% chosen option A, 67% B, 17% C, and 11% D. And in YouTube, everyone chosen option B. Okay. So what's the basic thing here, what we have to do? Good afternoon, Darshan. So the thing is, they are asking about nitrogenous waste excreted. Okay, nitrogenous waste can be excreted in 3 forms, right? We know that. It may be in the ammonia form, or it may be in a urea form, or it may be in the uric acid form. right? Where we normally see ammonia form. Ammonia form we normally see in the aquatic forms, right? And where we will see the urea form, just the regular mammals, right? Mammals normally excrete in the urea form regularly. Some of the fishes also, that's secondary. And uric acid, where you can see it. Okay, Sanjay, good afternoon. Uric acid you can observe in the birds and some of the reptiles, right? Their water conservation is more, right? Land snails, lizards, exactly. So the animal which want to conserve more water normally go for the uric acid. It's not intentional, okay? That's how it's made, not intentional. But in a general way, we can say like that, right? But the animals which already have plenty of water, which can dilute it, the things, so it will normally go for the ammonia. So there he, after observing the question, if you go for the options, here we normally come across with two things. One is the frog and another one is the tadpole. Okay, what's the difference then? Tadpole is a larval stage of the frog, right? So where tadpole is present then? Tadpole is present in the water. So it never comes out of the water because it don't have that kind of anatomical structures. But frog is present in the terrestrial, exactly, Preetham, right? It's in the terrestrial system, exactly. So you can compare the things, right? Here, even though you don't know, it's a direct kind of the answer. I agree to that, but you don't know it. Okay, still you can answer this. How? Tadpole is present in water, then you just go for what? You have to go for aquatic. That means ammonia for the tadpole, right? Just try to find it. Urea in tadpole, ammonia in tadpole, urea in tadpole, urea in tadpole. Everywhere they mentioned urea in tadpole, only one where they mentioned ammonia in tadpole, you can directly select this option. Okay. So what is the other one? Frog in terrestrial system. Okay. We will take care of this one. Urea in frog. Okay. Correct. Urea and frog, correct. Uric acid in frog, it's not correct. It's only in birds and reptiles, uric acid, right? And ammonia in frog, that's also not correct. Ammonia is excreted in frog, we agree to that, but the composition is very, very, very tiny. Okay, that means only 1 or 2% of the excretion will be happens through the ammonia. Most of the things will happen through the urea only. That's why we normally call frog as a uricotilic, not as the ammoniotilic. Okay, so which is the correct answer then? If you consider the thing, so frog is a terrestrial and it will normally excrete in the form of urea, and tadpole is water and it normally excretes through the ammonia. So this is what we explained or discussed in the last chapter, sorry, before chapter. So the correct answer is option B, urea in frog and ammonia in the tadpole. Okay, so remember the things regarding all these things. Okay, we will come to this one more time later. Second question. I think this is one of the easiest questions, right? Milk sugar is glucose, fructose, lactose, sucrose. I don't think anybody need any time to answer this. I'm just launching the poll. Some of you haven't read the answer, sorry, question. You can go back and answer later. That means within 1 minute. Okay. So I'm just waiting from the YouTube also. So, okay. Prajapati, I think you can also answer this question. So it's an easy one, right? So last question, yeah, I got the answer. Good. Last question, when you came, it's already going on. I almost ended. That's why. So where is Ashwin? Ashwin gave the answer. Anu gave the answer. Okay. I left it someone. Like someone is missing. I'm just thinking. Like I don't know. Okay, I'm getting almost correct answers from everyone. So last 10 seconds, guys. So we'll see. This is one of the easy questions, right? So everything is related there. That's why I'm ending the poll, guys. So only 69%. Why? What is other side doing? Akhil is missing. Akhil is missing exactly. Have to try to find. I'm ending the poll. I am sharing the results. 15% chosen option A, 6% B, 63% C, and 17% D. Okay, so most of you people chosen for option C, and some of you are having doubt regarding A and D. In YouTube, everyone choose an option C only. Okay, we'll see then which is the correct answer. The question is simple. They're asking about the milk sugar. Okay, so if you know, you can directly go. There is four options are there. Glucose is the simplest sugar, a simple sugar you can say. It's present in everywhere. Fructose is the fruit sugar, right? Fruit sugar. And sucrose, what's the common name for the sucrose? Can anybody tell me? Sucrose, common name. So fructose is a fruit sugar. Lactose is the milk sugar. Okay, I will write down. It's this kind of direct question, right? What's the common word for sucrose? Sugar. What sugar? What sugar? Fruit sugar, milk sugar. Anybody? You can say that. Okay, table sugar, but we normally don't use the term table sugar, but we will use the term corn sugar, or we'll also use the term another one. What is it? Why it is called corn sugar? Because sugar can be sucrose can be extracted from the corn syrup. Okay, corn syrup, and there is also from another one plant we can extract it, right? So most widely distributed plant, I think everyone know that. I'm just waiting for your answers. From which plant you will extract sugar? Exactly, corn sugar, cane sugar, sugar cane plant, right? Sugarcane plant, corn sugar, and it's also called as another one term is there. So beet sugar. Okay, why it's called beet sugar? Because it can be extracted from the beetroot. Okay, so that's why whenever you eat beetroot, you feel kind of sweet, right? A lot of sugar. So green sugar. What is that? Right. So that's it have a lot of names. So the secondary, right? But I think main course here, they are asking about the milk sugar. So that's actually the lactose. It's a direct question. Direct question. Okay. So you can also think about this one, right? Lactobacillus, so which can make the curd, and everything is related to lacto, right? Lacto, from where actually milk sugar, that particular word came from, right? You can think about all those things. Lactic acid, so everything. So it's related to lactose. You can think in other things also. Exactly, Prajapati. It's also called as the grape sugar. Okay, so exactly, so many names. So for the sucrose only, we will see. Okay, so what are the names and how the things, everything. Okay, so first one, we are discussing about the biomolecules. So what is biomolecules then? Okay, so why we use the term, a new term called biomolecules? We could have used the term molecules only, right? How it's different then? Okay, so difference is the biomolecules are the chemical compounds found in the living organisms that matters. Okay, so molecules present everywhere. It is present in the living and non-living things without any exceptions, or like without any bias in nature. But the biomolecules are the molecules which is present in the living organism. Okay, so what do they contain? They contain organic and inorganic compounds. Okay, so they contain both organic and inorganic compounds. Okay, so that's the second word where we discussing here. Okay, the biomolecules are the compounds, or the biomolecules are the molecules which is present in the living organism, and they may include both organic and inorganic compounds. Okay, you can see the structure here. So some of the structures, one is the amino acid structure, and is the protein structure, and another one is the some carbohydrate something structure. Okay, so different structures. Everything is chemicals only. Okay, chemical molecules only, nothing else. Okay, the first thing here is like how to check biomolecules are present in particular one, or how to examine the things. Okay, so or how it is different. Okay, that's the thing. The first thing, so way we normally compare is like, what's the difference between a non-living and living matter composition? Okay, when you compare the Earth's crust and the human body, as you can see here, right? So compositions of the elements are almost same, but the percentage of compositions are different. As you can see, hydrogen Earth's crust is present like 0.14 percentage. Its human body 0.5%. Okay, you may think that it's a very fraction, but according to the like how much composition we are made up of, it's actually a huge scale, right? Even a single digit make a lot of difference. Carbon only 0.03 in Earth's crust, but 18.5% in human body. Oxygen 46.6 in the Earth's crust and 65% in human body. Remember, we are talking about Earth's crust, not about the Earth's atmosphere. Okay, why we are comparing to the Earth's crust, not to the Earth's atmosphere? Because the living organisms developed from the Earth's crust, right? So it is developed from there only. That's why we are normally comparing with the crust, not with the core or with the atmosphere. Okay. If you go for nitrogen, Earth's crust it's very little. Okay. If you go for atmosphere, it's huge, right? So almost like 70-72%. But if you go for here, it's like very tiny. Nitrogenous components are very tiny in the Earth's crust. But in human body, it's almost like 3.3%. And sulfur is 0.03, but human body is 0.3%. That means almost tenfold. And sodium is 2.8, but in human body is very less, 0.2, but that's more than enough. Okay. And calcium 3.6, but the human body 1.5. Okay. Do you guys know? No need to remember all the things. I think. I will come back to that. Okay. And magnesium 2.1%, and here 0.1. Silicon 27.7%, but in human body it's highly negligible. Okay, normally not that much of silicon compounds are present in our body. Okay, so even though there's another debate is there. Okay, in the alien life, some people are speculating that the alien life may be a form of silicon also. So there are like speculations also in that way. Okay, so first question, like have your books have this particular table, like this particular table, is it present in your books? I'm just asking. Exactly, right. Yes. So I don't know about the old books, but it is in the new books, and probably most of your books have this particular table. Okay. So even though remembering all these composition is not at all important. Okay, but you have to know the difference between which one is present more in human and which one is present more in the Earth's crust. Okay, so then I just want to ask you as something. Okay, normally you know that we need calcium in your body, and somebody probably taking a calcium supplement tablets also. Okay, but what happens if you have more than enough calcium? Like if you take more calcium to your body, what will may happen? So it's devastating. Exactly. So what I'm telling here, this human body composition is the percentage of composition, and it should be in that way. Okay. If you try to increase any element in your body forcefully, okay, we will become Earth's crust. So probably, like that only. Exactly. If you try to increase any of the element in your body more than sufficient, then that particular person will die and he will become Earth's crust. Okay, exactly, Pawan. Okay, that's what happens. So these are the composition when we compare to that. Okay, so these are the percentage what we are running, and calcium importance, sodium importance, we will learn that later. Okay, and when we compare it to here, another one table is there. If you take a complete human body and compare it, then you can observe this hydrogen and oxygen, the higher percentage of hydrogen and oxygen mainly come from the water, which actually like fills 62% of our body, because every cell of your body is mostly contained water only. Okay, apart later everything will be mixed together. So next 16% is the protein, where actually nitrogen is involved. Actually nitrogen and carbon is involved. Okay, next carbohydrate is very less. Okay, so why carbohydrate is very less? Carbohydrate are only act in particular sites only, not like building the body. Okay, and these are like only mostly for the food purpose. And next is the fat. So 16%. Okay, similar to the protein only, because these are also storage materials, right? And minerals will be 6%. mostly in the bones and every part. Okay, all the others will be in the some fractions, some of them major fractions because of the water and everything, right? So different portions will be present also. Okay, how to separate these things then? To analyze the chemical composition, we have a method. Okay, what we will do in that method? We will take a plant sample, and then what we will do? We will actually grind this with acid compound, that is the trichloroacetic acid. Okay, TCA, or I can try chloroacetic acid, acetic acid. So after grounding this, okay, so we will get extractant. So as you can see, we have to like pour it through a cheesecloth, or we can say a paper. Okay, so after that you will end up with two things. One is upper pellet like things. Okay, and later there will be a liquid kind of thing. Okay, this liquid compounds is soluble fraction. That means what? So soluble fraction means the compounds which can be soluble in this TCA is actually completely made soluble in this particular acid, and it is present in this particular liquid. Okay, which are insoluble are formed these pellets. Okay, insoluble pellets. Pellets means a tablet like thing. Okay, insoluble pellets we can say. So this is how you can differentiate, how can I say, a soluble fraction and insoluble fraction. Okay, what is the importance of this then? Based on this, we can also consider this as the soluble fraction as the juicy substance. You can say that. Okay, this soluble fraction and insoluble fraction, and also as the different compounds. Okay, soluble and insoluble fraction also as the different types of the compounds. Okay, one second guys. Yeah, so this is how you can differentiate a molecule into two different kinds. One is the very tiny molecule which can be passed through, which are soluble and which can be passed through a cheesecloth, and some of them are not that much completely insoluble, and they will make a pellet kind of the product. Okay, so you can differentiate based on that as the biomicro molecules and macromolecules. Okay, so there is another way. How can you differentiate before to this a particular molecule as organic or inorganic? Organic or inorganic? So before that, we have another one question. What is the meaning of organic then? What's the meaning of organic then? Organic. Exactly. Presence of carbon, carbon. Okay. So, so many people are giving correct answers. Okay, naturally occurring, which having the carbon, Synthana, exactly correct, Akshay correct, exactly, okay, exactly. So there is one question I have. Okay, for the people who are telling that the carbon containing structures are organic, is graphite is organic? I just want to ask. Okay, is graphite is organic? Graphite. Some people are saying yes, some people are saying no. What about diamond? Diamond is organic or diamond? No. Exactly. Then, exactly right. So, what is organic then? The compounds which are hydrocarbons. Okay, hydrocarbons, exactly. That means carbon, it should be bonded with the hydrogen. Okay, so not only the compounds which are carbon, containing the carbon, the compounds which have carbon which is bonded with the hydrogen, then you can say it's organic. So some of you people gave the answer in the first phase only. So exactly correct. You can call compound as organic if it have a hydrocarbons. Hydro means this one, hydrogen with the carbons. Okay, so compounds with carbon with a bond with a hydrogen, you can call it as the organic molecules. Exactly. The other things what we are discussing here, like graphite or diamond, everything is allotropes. It's only made up of the carbon. It do not have any hydrogen or anything. So that will leave the other thing, right? What are inorganic then? Inorganic is the all other things. Without carbon and hydrogen bonding, all the other things will be inorganic. So this is how you can differentiate organic carbon and the inorganic carbon, right? So if I give you an example like alcohol, okay, so I will give you like ethyl alcohol, okay, or ethanol. So ethanol will be an organic compound. Everyone know that, right? And if I give you a carbon dioxide, okay, carbon dioxide, then the carbon dioxide is an inorganic carbon, right? Because in carbon dioxide, carbon is not bonded with any hydrogen. So can I differentiate that? So that is what become matters exactly. Carbohydrate. Carbohydrate is carbohydrates we will see. Okay, not like carbohydrate definition is little bit different. Not only what is that? So here we stated these are the what we stated simply about the hydrogen with the carbon, but carbohydrates are little different. There are like different functional groups will add on to that. Okay, so these are the things. Okay, so how can you make that? First you have to make separation between organic and inorganic, but both combine together and they will form compounds. In that compounds you can again differentiate between two things. One is the micromolecule and a macromolecule based on their size. Okay, based on their size. The first thing is the biomicro molecule. What are biomicro molecule then? The biomicro molecules are nanomolecules. These are having below 1000 Dalton. Okay, Dalton is a kind of a unit. Okay, exactly. This is like a measuring unit. Okay, it's a very tiny unit. Okay, Dalton is the tiny unit. So the molecules which have the weight of below 1000 Dalton, you can keep that in the micromolecules, and which are having above 1000 Dalton, you can call them as the macromolecule. Okay, the only exception here works is the lipids. Okay, because lipids normally insoluble in the sugar solution, insoluble in the acid solution, acid fraction. That's why we will keep all the lipids in the macromolecules only. Okay, but when you compare the other things, so we will keep the simple sugars in the micromolecule, but as the complex sugars in the macromolecule. So understood? So think about like this, there is a bead, then we keep this bead in a micromolecule, okay, and think that there is a chain which is made up of the beads, okay, then we keep it as the macromolecule. Why? Because this size is less than 1000 Dalton, but this thing is larger 1000 Dalton, right? So this is how we will make the differentiation. Okay. Yeah. First thing we are going to study in this is the carbohydrate. Okay. So what are carbohydrates then? Dalton means Dalton is the voice is breaking. I just okay. Oh, okay. I'm just opening the chat box. Okay, I will start answering from the one question. So Dalton is a measuring unit. We use it for the very tiny molecules. Okay, so until the way so these are very tiny, right? So it can't be measured in the any other units because it will be very like 10 raised to something will happen if you use that. That's why. Okay, and what is the scope of studying biomolecules? Best thing is understanding the basics is the to understanding everything, right? So biomolecules, understanding of the biomolecules, their composition makes us to understand what is happening in our body and around us. Okay, so biochemistry is majorly deals with these kind of things. Normally we will say something. Okay, when we are like going in BSc and MSc. So there are different kinds of books are there. Okay, but there's a book called Lehninger Biochemistry. Okay, normally I will say like if you complete the Lehninger Biochemistry, you're like you're kind of a god in biology because whatever the thing we are discussing, right? We discuss about the hormones, or we discuss about our expressions, or discuss about our thinkings, right? Everything is a chemical reaction which is going in our body, right? So these chemical reactions again is reason for the interaction of the biomolecules, right? If you know what that particular biomolecule is made up of, or what that particular compound is made up of, then you can understand these things very easily, right? So that's the important or the basics of biomolecules. If you eat something, how it is digested, everything we will learn that in the physiology. But from which compound it turned into another compound, how it is turning, everything we will study in the biomolecule. Okay, so in here you don't study anything at all. You only study about the like sum up or just a view only. Later in the chemistry, in the 12th class, you will study this in detail. Okay. Brown sugar is obtained from the plant. Brown sugar is the word used for the brown sugar. There is like two types of things are there. Okay, one is the regular plant obtained brown sugar. Sometimes for the cocaine also the people will use the term brown sugar. Okay, and chemical composition of I will come to that. Voice breaking. 1 second. Okay, so now my video is off. I think everything is okay now. So how to analyze chemical composition of organic and inorganic compounds? 1 second, then I will go back to here. Okay. So how to analyze chemical composition of the organic and inorganic molecules? So organic, first we will come to the inorganic. For the like study of the inorganic molecule, we will go for the analytical methods. That means we will dry a particular sample. It may be a plant, a root, or the leaf, or anything. Okay. Then we will burn that. When we will burn that, all the organic molecules, that means carbon molecules, everything will be it will because of the fire and everything, it will be burnt off, right? The only thing left in that particular solution will be the inorganic molecules. Okay, so then we can study about the things as you can study the qualitative analysis, right? Exactly doing the same thing, you will do that. Okay, so then come to the organic molecules. We can think about these are compounds are present, but we don't know the structures and everything. For analyzing the structures, then we have some other methods. Same, all these are chemistry, okay, not the biology. Like that's why it's like a midpoint from the biology and chemistry. So there, what we will do, we will actually analyzing the different reactions to the different compounds, we will come to a particular kind of the structure. Okay, so if it's a ring, or if it's a aliphatic compound or aromatic compound, does it have alcohol group? If you have the alcohol group, then it should react to this, and if it's an aldehyde group, then it should convert itself into a ketone, something, doing all those kind of things. Okay, introduction to both macromolecules and micromolecules can contain inorganic compounds. Inorganic molecule. No. Normally, when we are talking about inorganic molecules, that only confined in the micromolecules, because micromolecules may have the inorganic molecules. Okay, not the compounds. Okay, all the minerals, everything will be restrained in the micromolecule only, because this will be tiny, and macromolecule normally contain the polymer of the micromolecules. Simple, polymer of the micromolecule. Still voice is lagging. In the first class only we told, right, biology is mainly based on chemistry and also on the physics and the maths. So it's like combining everything, you will come across with this one. Okay, so 1 sec. What are the carbohydrates then? Carbohydrates, if you go for a definition, the carbohydrates are polyhydroxy, aldoses or ketosis, and their condensation product. A simple definition. Condensation products. Okay. What are aldoses and ketoses? Aldoses and ketoses. Aldoses are the compound which have the aldehyde functional group. What is the aldehyde functional group? Can anybody tell me what is the aldehyde functional group? Exactly, CHO. And the ketoses are the compounds which have the ketone functional group. Okay, so it will be C double bond O C R dash. Okay, so exactly. So aldoses are the compounds which have the aldehyde functional group, and ketoses are the compounds which have the ketone functional group. So what are the carbohydrates? Carbohydrates are the polyhydroxy. That means many hydrogen oxygen containing groups which have the aldehyde or the ketone functional groups. Okay, so these are the, one second guys. Okay, these are the basic thing about the carbohydrate. Okay, what is the general formula for the carbohydrate? For the general or the simple sugars, for the simple sugar, what is the simple sugar? A sugar with single unit. The common formula is CnH2n. One minute. CnH2nOn. Okay, you can think about the glucose here. What's the composition of glucose? Glucose is the C6H12O6. Right. For the simple sugar, the formula is the CnH2nOn. Okay. So how much of N carbon contain that? Sorry, double of the hydrogen will be there, and the same number of the oxygen molecule will be there. Okay. But for the complex sugar, a complex sugar, it will change. For the complex sugar, the particular thing will be CnH2O N. Okay, so it will be slightly different. Okay, it will be slightly different than that particular one. Okay, is it become both same? No, right? No, exactly. So it become both become one. CnH2 minus 2n minus 1 O n minus 1. Okay, so we can write it. The complex sugar, there will be CnH2n minus 1 and O n minus 1. It will be little different. Okay, so the general formula will be different. Why it will become different, we will see that later on. Okay, so why it's different, everything you will see that later. But the issue is like the definition is important. Okay, when we go for the words or the carbohydrates, we normally use the term, sugar or the saccharide. Okay, the word saccharide is also came from the word sugar only. Why have this particular name? Because these are the sweet in taste. We will tell you how you can differentiate a simple and complex. We will see that in the next slides. Okay, no issue. Simple means a simple one or two molecules will be there, compounds will be there. Complex will be having more compounds. Okay, so the saccharide word actually came from the sugar word only. Okay, so that's why we, instead of using the term carbohydrate, we will also use the term sugar or saccharide in a more common way. Okay, complex sugar. Okay. Start. And this majorly sweet in taste. If you can think about the sugar, what you normally eating, or you can think about honey, right? Everything is normally sweeter, but that's not exactly correct, because not all compounds of the carbohydrate are sweet in taste. If you eat this one, potato, it doesn't feel taste, right? It doesn't feel any sweet. It contains starch. It have that particular one. It have the carbohydrate only, but you don't feel sweet in that one. But if you eat jaggery or honey, you feel lot of sweetness in that. That's because depending upon the structural complexity, the sweetness maybe differs from the one group to the another group. Okay, so in that we normally differentiate it into three major types. Okay, based on This we normally differentiated the carbohydrate into a monosaccharides and oligosaccharides. Last one is the polysaccharides. Okay. So remember, wherever we are using the term saccharide, that means a single unit. Okay. Saccharide means a single unit of sugar. Okay. Based on these changes or these particular classification we made based on how many units are there. Okay, based on how many units are there, we actually differentiate into monosaccharide, oligosaccharide and polysaccharide. Okay, so we will move to this one. First is the monosaccharide. When you come here, you will try starting to understand all the things. Okay, monosaccharide means the compound which having the single sugar unit. Okay, so common example, as you can observe here, it may be a glucose or fructose or galactose. Okay, if you check this particular one, this particular glucose is a single molecule. Okay, it can be written in an open chain or like this. If you see the structure, so this is a complete one molecule or one saccharide molecule. So this is the particular one. So this is what you call it as the monosaccharide. Okay, so if you observe here, it is made of the 6-ring structure. In that one side you can find the oxygen, and from later on you can find the carbons. So from here we will start to name 1, 2, 3, 4 and 5 and 6, naming for the carbons. Okay, if you observe the fructose, you can see the difference in the structure. This structure is having only 5, this one, right? So this has the central oxygen, and carbon will be 1, 2, 3, 4, 5. So it's a 6 carbon atom compound. So you can also call this kind of compound as the hexose. Okay, and this the a 5 carbon compound you can call as the pentose. Okay, so these are the simple differentiation of the sugar, and there is a galactose. And if you observe glucose and galactose, both of them are hexose only, and both of them have a similar structure. Then what are the things which is changed here? The arrangement of the OH and H group make the changes here. Observe here in the first carbon atom, all of them are monosaccharide only, all of them are simple sugar only. Okay, simple sugar only. When complex sugar will come in the macromolecules, right? There we will discuss that. Here we will only discuss about the micromolecules or the simple sugars. Okay, so if you observe in the first carbon compound, so H is present in the outer portion and OH in the inner portion, but it's completely ulta in here. But other things will be similar if you compare to that. Okay, so based on like this kind of the things, monosaccharides can be differentiated to two things. One is the hexose and the pentose. Okay, and based on its aldehyde and ketose group, it can also divide into two types. The compound which have the aldehyde functional groups are called as aldoses. Okay, the example of the aldoses are ribose sugar. These are the name of the compounds. Okay, this one, structure is not there, so I'm not going to write that also. And glucose. Polyhydroxy is multiple hydrogen and oxygen containing structures. And galactose. Okay, glyceraldehyde. Glyceraldehyde is the simplest monosaccharide or simplest aldose. Okay, and the compound which contain ketone groups are called ketoses. The examples are dihydroxyacetone, ribulose, fructose, xylulose. So you can observe here is also. So in the glucose, it has the CH2OH composition, which is making the aldose groups normally present in the outward portion of that particular compound, but ketosis will be present inside portion of the things. Okay. So, but remember this some of the examples which are very important. Okay. So the examples will be aldoses, ribose sugar, glucose, galactose, or glyceraldehyde. For ketosis will be dihydroxyacetone, riboses, fructose, and xylulose. Okay. Based on number of carbon also, the monosaccharides are divided into several other things. Okay. If you take example of glyceraldehyde, okay, if you take an example of a simple glyceraldehyde, once again, guys. What about erythrose and what are those numbers on glucose and photose? The numbers representing the number of carbon atoms. So which number of carbonate is present? That is what it's actually representing. So these are the numbers. Okay. These things, erythrose and another one thing is glucoheptosis. So heptosis normally representing the ketone groups, and another thing erythrose also representing the ketones only. But I thought that that's not so important. That's why I left it. Okay. There is only one carbon atom in glucose. No. Sorry, I will try to write in this way. So glucose can be written in this way. So based on this, I told you, right? Glyceraldehyde, we can write the simplest one, glyceraldehyde, or we can take the glucose is the structure. The structure will be like this. Every point you have to write the carbon. Carbon is not mentioned in here because it's understandable, because it's the base chain, right? Probably Shiva sir already told about those things. When you are writing those structures, we will normally left out the carbon this ones, because it's like assumptions. We can make the assumption. That's why. So one carbon, and here is another carbon with the CH2OH. So every one representing the no 1, 2, 3, 4, 5, and 6. Everything is representing that particular one. Okay. So highest one is the heptose. Highest one is the heptose. Normally we represent up to that only. Okay, I'm just rubbing this particular structure of the glucose, because you can only represent in below form only, right? I don't think anybody have that particular problem in understanding that one. Okay, so I just want to write the glyceraldehyde. That's why. Okay, glyceraldehyde, you can write it in a simple way. So carbon with the C double bond OH, that means aldehyde group, and with the OH and H and CH2OH. Okay, so based on that, how can you see that? So you can write it as the triose. Why it is triose? Because it has the 3 carbon compound. 3 carbon compound, right? So example will be acetaldehyde, glyceraldehyde. Sorry, not acetaldehyde, glyceraldehyde. What you call something which have the 4 carbon atom. So we call it as the tetroses. Okay, tetroses. So you can think about the triose or erythrose, all those things. Okay, and based on if it is having the 4 carbon compound, and if you're having the 5 carbon compound, and you call it as pentose. Pentoses, that means 5 carbon compound, and if it contains the 6 carbon compound, then you can call it as the hexoses. Okay, hexoses. And if it contains 7, then we call it as the heptoses. That means 7 carbon compounds. Okay, the only issue matters here, like more than 7, I think there is some structural integrity problem will happen. That's why normally we'll it will be only up to the 7. Okay, So you can differentiate in this way. Remember, a triose is also a monosaccharide and a hexose is also a monosaccharide. But the number of carbon, it differs in every unit of that one. Okay. So what is happening here? A triose is also a monosaccharide and a pentose, hexose or a heptose also a monosaccharide. What is monosaccharide? Monosaccharide is a single unit of the sugar. Single unit of sugar. Single unit of the sugar. One second. Yep. Single unit of the sugar. Okay. So never represent monosaccharide based on how many carbon is there, because number of carbon may vary within that particular unit. Okay. You have to represent, or you have to look through that particular what we can say, how many number of units are there, not based on how many number of carbon is there. Okay. No, it came under micromolecules. So please mention whether they are keep it in front of every monosaccharide. Just you can look about here, right? So you guys are talking about triose, tetrose, pentose, hexose, and heptoses. No need. Remember, this triose compound may be a aldose or maybe a ketose, right? So if you take here glyceraldehyde, you can call it as a triose, right? And exactly dihydroxyacetone is also a triose, because both of them having 3 carbon only, but the functional group is different. But here glyceraldehyde, it have a aldehyde functional group, but in dihydroxyacetone it have the ketone functional group. Okay, so that's the only difference. And regarding glucose, fructose, galactose, you can check in here only, right? Aldosine ketose list where it is represented. So remember, the triose, tetrose, pentose, hexose, heptoses classification only based on number of carbon which is present inside that particular compound only, inside that particular unit of the sugar, not based on complete thing, based on that particular unit of the monosaccharide. Okay, can I rub off these things? So I think so there are like one more thing will come here. That's why. Okay, we will leave it. So next, so something else came. Okay, all these monosaccharide, we call them as the reducing sugars. Okay, we call all this monosaccharide as the reducing sugars. What is the meaning of reducing sugars then? These monosaccharide have one ability. Okay, what is that ability? This monosaccharide can convert or can reduce Cu2 plus ions into the Cu plus state. Okay, these sugars, all of the monosaccharides have the ability to reduce Cu2 plus ion to the Cu plus state. Okay, Cu2 plus state to the Cu plus state. Okay, so reduction is happening, right? Why reduction is happening? Because of the exposed aldehyde or ketose group. Okay, the exposed aldose once again, exposed aldehyde or ketose group reacting with copper compounds, and it's converting or it's reducing Cu2 plus state to the Cu plus state. That's why we call all monosaccharides as the reducing sugars. If you check this one, this is very important in the biochemical machinery. Okay, what's the reason? Because of that, you can find out the percentage of monosaccharide present in a blood or in the urine using this reducing sugar property, reducing property of this monosaccharide. Okay, there is a test called once again, guys, in the notes I will keep this in the different page. Okay, no need to take tension. So we normally go through a test called Benedict test. Benedict reagent is compound which having the Cu2 plus compounds. Okay, so when it's treated with the urine, so depending upon the percentage of that particular sugar present, it will give the different colors. Okay, blue color is the color of the Benedict reagent. That means if there is no reducing sugar or no monosaccharide is present, then it will give the blue color. If it is like some percent, right, 0.5 to 1 gram is present in that particular urine sample, then it will give the green. And if it is more, it will give yellow. If it is still more, it will give orange. And if it is high, then it will give the brick red color. So based on the test result or based on the color of the Benedict reagent, so we can come to the conclusion how much sugar which is present in the urine of the patient or the blood of the patient. Okay, these are the things. So we'll move on for a little bit. Second one is the oligosaccharides. What are oligosaccharides? Oligosaccharides means oligo means few. Okay, oligo means few. Saccharides means sugar. Oligosaccharide means these compounds have 2 to 9 units of saccharides. Here, remember, I'm not talking about the carbon. I'm talking about the unit. Okay. Based on that, it's also divided into several types. That means, how can you differentiate? You can differentiate into disaccharides and trisaccharide, tetrasaccharide, pentasaccharide. Right, exactly. But the importance are the disaccharide. All the other things are not so important. Right. I'm simply writing it for your reference. Disaccharide, trisaccharide. Remember, last time you used the term triosus, tetrosus. Here we are using the term disaccharide, trisaccharide, tetrasaccharide. Okay, tetrasaccharide and pentasaccharide until what? Until up to the 9 sugar unit or 9 saccharide unit. Okay, so in this, disaccharides are important one. All the other things are not at all in the syllabus and not so important for our studies also. Okay, how these disaccharides or oligosaccharides will form? This disaccharide or oligosaccharide will form by joining of the monosaccharide. How it will form? Joining of monosaccharide, or condensation of the monosaccharide. So just imagine this thing. So we'll take a very simple example of the sucrose. A glucose and fructose will be present. Okay, so they actually omitted the H also. That's why I only written OH. Don't think that where H is went. Okay, H is there, but they omitted it while writing it. Okay, it's assumed. So what happens here? Regular structure of the glucose will contain one OH and another one H here, right? If you observe the last page, so we will back to the here. Glucose contain OH and H, and galactose contain completely opposite to that. So here will be H and another portion. Sorry, I'm not writing in this one. We'll make use of the which one? We'll take use of this one only. No problem. OH and H, here you can find the another H. Okay, what happened in here then? So while forming the bond, it will lose a portion of its bondings. Okay, portion of its bondings, and it will form a bond called as the glycosidic bond. Okay, I will write it down one more time here, how the bond formation will happen by using another one thing. 1 second, guys. Okay, so I'm writing the regular sugar or regular glucose here, H and OH. All the other things are unnecessary for us. CH2OH. Okay, we will take another one glucose molecule only. Okay, so CH2OH. And here what happens? H and OH is present. Okay, while bond formation, what is happening? A portion of the H from here, OH from here, it's got removed. That means condensation is happening, and it will give rise to this particular molecule. And along with what? Along with the removal of the water. That means condensation is happening. That's why water is getting removed, right? So it is forming a bond. This bond is called as the glycosidic bond. This bond which is happening in between these two are called as the glycosidic bond. Exactly. Trisaccharide is it by joining? Exactly. So wherever here oligosaccharide 2, exactly. In here, when we are discussing disaccharide, I told you, right? So what is happening? Two monosaccharides are joining and going through a hydration or condensation process. Condensation means they are combined together and they are forming, or the bonds in between them, in between the carbon and the another one oxygen groups, by leaving the water as the byproduct. This is called a glycosidic bond. This is how they will make the disaccharide. If three monosaccharides will join, then it will become the trisaccharide. If it is more will join, four will join, it will become tetrasaccharide. A five will join, it will become pentasaccharide. Okay. Exactly. These structures are important. You have to remember about this structure. Okay. You have to remember about this particular structure. That's important. Uh, what is actually condensation? The reaction which will leave the water, we call it as the condensation reaction. Okay, the chemical reaction which leave the water as the byproduct, we call that kind of reaction as the condensation reaction. Okay, so this is how it's forming. Is disaccharides exist? Exactly, disaccharides exist. Everything is exists. What we are studying here. Okay, biology is only study about the things which are existed. Okay, chemistry and physics, math will also study the things which are not existed, right? So we are studying about the biological organism. So whatever we are studying, it's existed only. Okay. Mm, huh. Hmm. Exactly, Chiran. Okay, yeah, we will see what happens, everything. Okay, so major examples for the oligosaccharide, especially disaccharides, are sucrose, lactose, and maltose. Here, remember, the sucrose is formed by the combination of the glucose and the fructose. Okay, and lactose is formed by the combination of galactose and glucose, and maltose is formed by the combination of glucose and glucose. Sucrose, you can call this particular structure as the this one. What we mentioned, right? Cane sugar and all the different cane sugar or table sugar and everything. And remember, this is the most frequent one or most producing one kind of the sugar. Lactose is the milk sugar, which normally present in the milk. Okay, this is one of the less sweet sugar, less sweet sugar, and maltose will be present in the germinating seeds. Germinating seeds, and sucrose will be present in the cane plant. Okay, and this one of the sugar we normally use it. Okay, milk of all mammals. Lactose is present in all mammals, sorry, all milk. Okay, it is present in the all milk. If you observe here, what happened, this particular compound is, so in sucrose, the both aldehydes and ketone groups are actually formed or participate in forming this particular compound. So glucose is the non-reducing sugar, but lactose and maltose, both of them are the reducing sugars. That means in the monosaccharides, all of them you can simply call it as the reducing sugar. But in oligosaccharides or in the disaccharide, you can't state like that. Disaccharides are normally reducing sugar, but not the sucrose. Okay, sucrose is the normally non-reducing sugar. Okay. Uh, These structures are not given in the book. Remember, this can be asked in the NEET. I think some structure is given. I'm not sure about which is given. Okay, but because all the structures you will study in the class 12 biomolecules chapter. Okay, there you will study about the structures. That's why you have to like started to study these things from here only. That's why you will not face any difficulty in there. Okay, I will revise one thing after this particular question. Okay, so we'll just like solve this problem. Sucrose is a reducing sugar, non-reducing sugar, both reducing and non-reducing sugar, and none of the above. I think it's too early to ask this question because now only I mentioned. No problem. Okay. You guys have like some 35 seconds. Okay, last 15 seconds. I'm ending the poll in 5 seconds. I'm ending the poll and I'm sharing the results. 27% chosen option A, 63% B, 11% C, and nobody chosen option D. Exactly, and in YouTube, everyone chosen option D. Correct. So the correct answer is exactly sucrose is non-reducing sugar. Okay, non-reducing sugar. I will glance up a little bit. The issue is like in your textbook, there is nothing given regarding this carbohydrate basic structures. They directly went to the polysaccharides, right? They given very tiny thing about the things. Like there's not that much of discussing in the biology biomolecule chapter because everything you will study in the chemistry biomolecules chapter only. Okay, this is a glance up. So we will make one more time from the initial things what we discussed. Okay, the first thing what we have discussed about the biomolecules are the chemical components which in the living organisms, and this may include organic and inorganic compounds. And this when you compare the percentage weight of the compounds or the elements which is present in Earth's crust and human body, you will come to a difference that the only major thing is the carbon and oxygens are present more in the carbon, oxygen, and hydrogen are normally present more in the human body or in the living state, right? So how can you differentiate the things? You can go for the acidic test. That means using the TCA or trichloroacetic acid. You can dissolve the things, plant matter, and you can separate the micro and macromolecules based on their solubility and how can they filtrate, right? How can you differentiate an organic and inorganic compounds based on their structure of carbon and not having the having carbon with the hydrogen, you can call it as the organic. Without having that, you can call it as the inorganic, right? So next thing is micromolecule. In that we are going to study about the carbohydrate. We have studied about the carbohydrate in the first phase. Carbohydrates are the polyhydroxyaldoses or ketones. That means many carbon hydrogen combining groups of compounds having either aldehyde as a functional group or ketone as the functional group, and it is also having their condensation product. That means when they combine to form some other products. Okay, carbohydrate can be given two general formulas. One is the CnH2nOn, that's for the simple sugar, as you can see in the monosaccharides. Now you know the word monosaccharide, so I can use the term, right? So that's the common term we are using. But there is another one general formula, that means CnH2n-1 for the complex sugar. Why it is that? Because during the formation of the complex sugar, due to the condensation reaction, water is get removed. That reduction in the water will reduce the number of the number of molecules or number of hydrogen or oxygen in this particular compound. Okay, the saccharides are the single unit of the sugar. That's what we are going to discuss. And based on number of saccharides or number of sugar units, we divide it into monosaccharide, oligosaccharide, and polysaccharides. So first thing is the monosaccharide. Monosaccharide means single unit of the sugar or single unit of the saccharide, right? This may be this may contain a aldehyde functional group or it may contain a ketone functional group. The common example for aldehyde functional group having sugars are ribose sugar, glucose, galactose, or glyceraldehyde. Glyceraldehyde is the simplest aldoses or simplest aldose carbohydrate. And ketoses containing sugars are dihydroxyacetone, ribulose, fructose, and xylose. Here, dihydroxyacetone is the simplest ketone containing monosaccharide. Okay. Sugar, simple sugar. What only monosaccharide, sugar, simple sugar. So all the monosaccharide and oligosaccharides are called as simple sugars because their size is small. Okay, here some of the examples. As we can see in this particular one, we are not at all discussing about the number of carbon present in it. Instead of that, we are discussing about how many how many units are there. Okay, here we are studying about monosaccharide. That's why a single unit is there. Based on the number of carbon present in the unit, again we are divided into a triose, tetrose, pentose, hexose, or heptose. These are majorly dependent upon number of carbon which is present in that particular saccharide. This triose may be a aldose or maybe a ketose. Same thing with all the other things also. Okay, and these monosaccharide are called as reducing sugars because the open groups, open aldehyde and ketone groups, have the ability to reduce the Cu2 plus ions to the Cu plus ions. So these will be helpful in detecting the monosaccharides in the different solutions. How it will do that? We will use two different reagents. One is the Felling reagent, another one is the Benedict reagent. So that we study in the chemistry practicals. Okay, using the Benedict reagent, when we test for this reducing sugar, probably Benedict reagent you guys use it for the aldehyde group also. Okay, so when we treated this with the sugars, then what happens? Depending upon the percentage of reducing sugars, the color of the Benedict reagent will change from the blue to the red. Depending upon the intensity of the color, we can represent how much percentage or how much gram of the monosaccharide will be present. Okay, when two monosaccharide will combine, then it will form the oligosaccharide. Okay, so what happens oligosaccharide when you join the monosaccharides, then it can form a condensed form. It can give rise to a condensed form. So that's what we call it as the oligosaccharide. Oligo means few, saccharide means sugar unit. That means a few sugar units combined form. Okay, oligosaccharide may contain from the 2 saccharide units to the 9 saccharide units. Anything below 2 saccharide unit is the single saccharide. That's what we studied, monosaccharide. Anything above 9 units, we call it as the polysaccharide. That means many saccharide. Okay, in this oligosaccharide, the important is the disaccharide. That means the sugar which having the 2 saccharide units or 2 sugar units. Okay, so here what happens, when 2 sugar molecules come together, they will form a bond by losing water. That's what we call it as the dehydration or condensation reaction. That's what gives rise to a glycosidic bond. Okay, glycosidic bond is a type of ether bond. So what happens, it normally happens between I forgot to wrote that down. Okay, so it happens something. So what happens? It will form a bond. Based on number of composition and where it is present, we normally have three important kinds of the oligosaccharide. One is the sucrose or the regular sugar, so which can be found in cane plant or in the sugar beet plant everywhere. And second one is the lactose that can be found in the milk. And third one is the maltose, where you can find in the germinating seeds. Normally use the term malt, right? Ragi malt or rice malt. Sorry, not rice malt. Finger millet malt powder and everything, because they are having the germinating seed powders, the main oligosaccharides that we are discussing. Okay. There are one or two things left. I think it's not important. If it's important, before tomorrow class, I will mention it one more time. Okay. So what is, I think like I will cover the things important. If I think anything left out in the saccharides, when we are discussing about the polysaccharide, we will mention it one more time. Okay. Can you break the bond by hydrolysis? Exactly. When you break the bonds of the disaccharide, then what happens? It will get separated by taking OH and H, that means by the hydrolysis exactly, and they will form monosaccharides. That means whenever you will break any disaccharide, it will give rise to the monosaccharide. Okay. What only next? Exactly. What is the meaning of reducing sugar? Okay, reducing means which can reduce the Cu2 plus ions to the Cu plus state in the Benedict reagent. We call it as the reducing. Okay, reduction. In oligosaccharide, sucrose is non-reducing sugar because the aldehydes and ketone groups are participated in the bond formation, but other lactose and maltoses are the reducing oligosaccharides. Okay. Oligosaccharides are the compound which have more than one sugar unit. Okay, it may have 2 to 9 sugar unit. The simple thing that we are studying is the disaccharide. That means having 2 sugar unit. Okay, so it will make a bond by losing the water. So it will form a condensation reaction. It will go for a condensation reaction and it will form a kind of ether bond. That's what we call it as the glycosidic bond. Okay. So normally oxygen will be in the middle. Sometime nitrogen will be in the middle. Okay. That time we call it as nitrogen will be in this side or middle. Then we call it as the N-glycosidic bond. Probably you heard the term N-glycosidic bond in the when we are studying about the bacteria, right? It is 12th book figure. All these structures are given in the 12th chemistry textbook. In the 11th biology, there is nothing to think or study about the structures or anything. We only study about the general characters and how it's made and everything. Only few important structures are given. Okay. But you have to remember all those things to understand the things. Okay. Okay, guys, like I'm just launched the teacher and peers, and okay, so tomorrow also we will revise this. You also read this thing one more time. I will make a few different kinds of the slides, and today also we will revise the things in the initial. Then we will go for the other things to study. Okay. So if there is any confusion, please put a message in the WhatsApp or in the Telegram. So I can also like put a graph or something picture to that. Okay. So how to draw structure of the glucose in the initial I shown you, right? Where is it? There is nothing even regarding the carbohydrate. Some cat is listening to my class. It's sleeping actually. This is the structure of the glucose. Galactose, you can find it in the animal body. Thank you. Okay, Sanjana, we will do one thing. I will add another one slide tomorrow. Okay, so join tomorrow class exactly at the time. So I will discuss that and I will go to the next one. Okay. You can ask her, Shushma. Thank you, Brahmaya. I think biomolecule chapter, this one, modules are already with you. The cell module only contain this also. Okay. So just check with that one time. Thank you, guys. in YouTube also. Okay, guys, we'll meet. Okay. Then I will close this one. Why all ozones are present in the stratosphere, on not in the layers of the atmosphere? Because of its way, I think. Okay. But ozone can be formed here also. But if ozone is present in the regular atmosphere, it's very hazardous. Okay, it's very problematic. Most probably due to its weight, I think. I'm not sure about that.